Olá! A IUPAP, International Union for Pure and Applied Physics, está interessada em muitas coisas. Dentre elas, a definição das unidades básicas que nós usamos no dia a dia, a manutenção das constantes fundamentais, mas também está muito interessada nos desafios que a ciência tem atualmente. Na área da física são muitos os desafios. Nós temos desafios desde o entendimento das partículas elementares, da matéria condensada, na física nuclear, na biofísica, na astrofísica. Em todas as áreas, ou sub-áreas da física, nós temos desafios. Para que você saiba quais são esses desafios, a IUPAP organizou um workshop especial, que chama-se Entendendo os Desafios da Física Moderna. Cada área da física está aqui sendo apresentada por um líder, membro da IUPAP, e que você terá então agora a chance de entender, através desta apresentação, os desafios que a área apresenta e como os físicos, de um modo geral, estão superando esses desafios para que a física seja um instrumento de entendimento das ciências naturais e que auxilie o homem não apenas a avançar o seu conhecimento, mas a tornar a ciência um instrumento útil da melhoria de vida de cada um existente nesse planeta e nesse universo. Assista e também faça parte do entendimento dos desafios da física moderna. Boa sorte!
The next talk is biophysics, Aihua Xie.
Good afternoon. Well, this is quite a different talk than I used to be, and it's also interesting to prepare and takes a lot more effort. So with biological physics, everybody knows about Schrödinger, but perhaps not many knows his contribution to biological physics. So, unlike other fields, I think biological physics, somehow many people are still uncertain what it is. So we start, what is the physics? Okay, and this is the definition I got from Wikipedia. And the biological physics, you basically change from a matter to biological matter. And that can be pretty complex from very different varieties, from molecular level to cellular level, tissue, and even higher systems. So basically, we can break down the question of biological physics into two. One is we apply known physics to understand biology. But there's also others, is biology can contribute to physics. And this is something many probably haven't recognized that. Okay. So early on, and the biological physics has already attracted those very bright minds, and Schrödinger and P.W. Anderson. Both of them expressed the opinion that the new physics to be expected in the organism, living organism. So the idea is basically the physics we developed based on the system we know, and not necessarily could explain everything in the more complex systems like biology. So Schrödinger in 1940s and the 1950s, he really made a big effort to think about biology. And he delivered two series of public lectures, and each lead to a very small booklet. So one is what is life, and the second is mind and matter. And so this writing after the lecture was published in the booklet of this What is Life. And this book has been reprinted for almost 30 times. And you still can't find that. It's still in print, okay? And later these two are combined into What is Life and with Mind and Matter. And this is a really very interesting book to read for physicists or non-physicists. And this book has inspired generations of physicists. And I will come back on this point. So what Schrödinger wrote in his book is when he is thinking about what is life, he is focused on this thinking is how can the events in space and time which take place within the spatial boundaries of a living organism and be accounted for by physics and chemistry. So that keeps him focused. And Schrödinger also commented the biological materials, like a genetic material, as aperiodic crystals. And he said, well, in physics, we normally deal with periodic crystals. And to physicists, and they are very interesting and complicated. But compared with aperiodic crystals, they are rather plain and dull. Well, this is illustrate why the aperiodic crystals are interesting. And you see our genetic material DNA has only 4 building blocks. And in our human genome, we are made of this 4 billions of this DNA pair. If they are periodic, they will not contain a lot of information. Because the nature they are aperiodic, so they can contain the old information to build a life as complex as human beings. And also for the aperiodic matter, proteins, the building block is 20 building blocks. And for even a very small protein of 100 amino acids, so the proteins can be up to 1,000 or more than 1,000 amino acids. So this 100 is really small. But since each can be one of the 20 amino acids, so the variety possible through this aperiodic matter is huge. And this gives the really the proteins has a huge options to evolve to be functional and function in many different ways. And the impact of Schrödinger's book, What is Life? And one very good example is about the double helix DNA. So one focus of Schrödinger in the book is he said he wrote that book, and one of the things really drove him is to say, at the time of 1943, they know some of genetics and mutations, that you inherit properties, but we still do not know what exactly are the genetic material and what are the mechanism of that how the information in the parent are passed on to their children. So in the book, it discussed about that so much information must be very compact. So he expects to write in the molecular format, like aperiodic materials. And so he asked the question is, how does this work? How it is possible if you have the genetic material, how can pass to the next generation? So that inspired the physicists, physicists and the non-physicist biologists, okay? And they worked together, and also through the chemists doing the crystallography and the defection, and they came up with this double helix structure for genetic information. So the key of this is called the base pair. You see these two, T and A, has 2 hydrogen bonds, and the G and the C has 3 hydrogen bonds. The energy of a hydrogen bond is large enough, much higher than the thermal energy, but still it's not too far away, so you still can break the hydrogen bond. So it's a very nice way the biology used this energy, okay? So because of this, so the A is always with T, and the G is with C. And this is the mechanism how the gene is copied for the next generation, because you just split the 2 into single strand, and then each one, there is only one to be. If you have T, this must be A. If you have a C, must be G. So you would get the exact copy of the DNA. So once this structure is determined, the mechanism is very obvious. So since this is one of the most significant discovery in biology in the last century, and the physicist has a role to inspire this discovery, and plus this Crick himself is trained in physics. So in the United States, there was a very clear two phase, okay? Before the year of 2000, the American Physical Society largely regarded the biological physics as not physics, okay? So you would not really see hirings in this field. And so one of a very talented physicist, he got the PhD in MIT and the postdoc in Harvard. And he was so smart, he knew he could not get the job in biological physics, so he studied statistical physics. And he was so bright, he hired as a tenured professor at the University of California, San Diego. As soon as he was hired and tenured, he said, now I switch to biological physics. And he is doing very well, Terry Huai, yeah. And, but in the year approaching the 2000, as the new century and the physics community is looking for opportunities, what are the best directions for biological physics? And then they found out biological physics. And so there's a Physics Today articles. And so at that time in the years, many physicists was looking for ways to enter biology and the biological physics, but they are not sure how to do that, okay? And they also do not know what they really can do. So American Physical Society organized a series of workshops. It's called Opportunities in Biology for Physicists. I was in one of the steering committee because I did a PhD in physics and biophysics, so I'm already in biological physics. And and this workshop, and that is one of the best workshops I have attended. We have both physicists and biologists. And this biologist said, biological systems are physical systems that obey the physical rules, so they should should have more, they may have more opportunities for physicists than for biologists. In a way, it is quite true. As I interact with biologists and physicists, and when I talk to biologists, they like to emphasize their system is very special. So they are not looking for general principles. But for physicists, we always had this tendency looking for general principles. And plus, we can combine physics and biology to make predictions. So really talk about what is biological physics, and I think that the area is field is very broad. So after the 2000, and the biological physics in the United States is rapidly growing, and almost every department is developing biological physics. And this is the several titles in one of our IUPAP conferences. So it's from proteins to DNA to membrane to system biology to cancer to soft matter, and it's quite broad. And certainly in this short session, I'm not able to, and plus I do not have the knowledge of such a broad area. But I would highlight a few recent developments. The physics is very important, and those technology development lead to as important tools in biology and lead to Nobel Prizes. So both prizes are related to this, is how to break the diffraction limit of conventional optical microscopy. Because when we want to study biology, the first is you want to be able to observe. Otherwise, you are just guessing. So something we want to observe at the cellular levels. Okay, so what are the inner makings of the cell and how they change during the in their living process, and others at the molecular levels, like proteins, DNAs, and RNAs. So the one technology is called the super resolution fluorescence microscopy. And as we see, the diffraction limit, even for UV light, is about 200 nanometers. And that is way too large than the need to see the inside of a biological system. For example, one is called a microtube, and that is about 20 nanometers. So this is the technology of a conventional microscopy. You can see very blurry, and that is resolution is about a fraction of a micrometer. And this is a super resolution. So how it is possible to break the this diffraction limit? So this is over the many years of study, and they using this blinking. So basically, they excite the fluorescence at very low intensity. So it's only one molecule in the diffraction region would have fluorescence. Okay, and then they repeat that over time, and they're doing a lot of data processing. And this technique, so it is able to make so small, and by also attacking, using this different fluorescence probe, and now in cell molecular biology to see how different parts of things interact, or DNA expression. So this technology plays an important role. And plus, and this field is continue developing, and they keep getting better and better microscopy for the applications of super resolution microscopy in biology. So this is a few years ago, the Nobel Prize in Chemistry, and he is Eric Batazik, is a PhD in physics. And when I was a chair for the American Physical Society Division of Biological Physics, And we give him, I was not in the prize committee, but the Division of Biological Physics gave him the surprise, very prestigious prize for our division, and shared with another professor in Harvard. So I told him about this, but he rejected. And so a few years later, he got the Nobel Prize in chemistry. And the second, I'd like to say is that, this technology has more to do in physics than chemistry. But this Nobel Prize is in chemistry. Second technology is more recent. It's actually just announced about a month ago. So this cryo-electron microscopy. As you see, also quite a physics. So knowing the structure of a protein is always very desirable. Okay. So this is the people to study protein structure function. They want to know the structure. And this is, we mainly show that the folding of the proteins. So these are rather small proteins. And this is the myoglobin in our muscle and the hemoglobin in our blood. And this the visual rhodopsin for our vision. But we have much larger systems. So how to study the structure of very large system and without crystallization? So these are the several structural technologies, and each lead to several Nobel Prize in each. So one is X-ray crystallography, this nuclear magnetic resonance, and this cryo-electron. So the advantage of this is you get a good structure, but they need the crystals, which is very difficult to get. And the solution AMR, you do not need a crystal, but the size is limited. The CryoEM, and that is, they do not need a crystallization, and they still had a really good atomic resolution. So this technique is, using this, the sample preparation is really a single molecule, okay? And then using electron beam, and then it was a rotator orientation. The challenge in this technology, one is the radiation damage from the high energy electron beams. And the second, the living systems need water. So how to solve these two are the really challenge. So this technology has been really developed over almost 50 years. So now this technology has a breakthrough. So you see, this is a very, very large system, and that is the ribosome, and that is the very big system with RNA and the protein, and that is to make where it is machines to make proteins. It's needed for every living systems and very important. So this is would be, for crystallization would be really, really hard to do. And this is another very large system. It published, and this work was expected to have a huge impact in biology with near atomic solution. This is to change the genetic information during the expression of the genetic information for the cell. They need changes in genetic information. So, and now this technology is gained application in many biologies, make things impossible become possible. So for this work, and they shared the Nobel Prize this year again in chemistry. As you see, the technology has more to do with physics than with chemistry. And this Johann Frank, he is a PhD in physics, and this Henderson and this Dobok, they also have a training in undergraduate in physics. And I know in the, how much time do I have? Okay, so I need to, okay, I just finished very quickly, okay. Sorry about this. So let me just skip that about the gaps in understanding in this. We need to know the dynamic structures. We need the sensitivity to protons and electrons. So we still need the new technologies in structural biology. Plus, with DNA is expressing so fast, so we got a huge number of proteins, but we do not know what they're doing. So this also leads us, we need to understand the principles in protein function, like Newton's law in physics. I would skip some of the things and go to the very end about the challenges. Okay, so the challenges in biological physics. So this field is still very, very young. We have lots of open questions. In a way, it's a great place for new opportunities. You can, other is the research funding. Like in American Physical Society, the physics division of and the National Science Foundation has a very small funding for biological physics. So we mainly need to get the funding from biological part, chemistry part, not from physics. So physics is still not supportive enough. And we still need better technologies. And the new physics, new physics, okay? So how to understand the complex systems. As one point I want to emphasize is in biology, when we deal with such complex systems, so currently, the most of research still is very localized. One lab doing one thing. But in order to address the big questions in very productive way, this international collaboration would be very important. In this sense, the IUPAP may play a role. And also, I'd like to take this opportunity as this international conference. Some countries probably still do not have a reasonably sized biological physics yet. And I really encourage you to think about that and get the book of What is Life, and to not miss this great opportunity for development and discoveries. Thank you.
Question? In the back.
Aihua, what sort of international collaboration or large-scale projects are you thinking about?
For example, at the protein levels, okay, and we do from preparing sample to experimental measurement and to data analysis and to so in the process, I can use my own examples. Like I'm developing infrared structural biology for dynamic studies of proteins. And then we would like total chemical synthesis. Instead of using bacteria to make proteins, if we can just using chemistry to make proteins. So we can make a bacteria to make okay. So different, basically, different group have certain expertise. If we can make, combine those expertise, we can make much faster progress. Others is equipment. Like the equipment in our lab is very, very rare. But it's difficult for every lab to get such equipment. So the access to the instrument and the access to expertise, I think those would be very helpful.
Okay. One more question.
Thanks very much. I'm interested to see electron microscopy. Having used semi-empirical modeling for understanding large proteins and knowing the dangers of computing models. Do you think that there is a new kind of the codes that's possible at the moment?
Actually, this is a very, very good question. There is another exciting field in biological physics. It's called the minimum data assisted structural prediction. So as the computer is getting faster and faster, it can do more for large systems. But if you do not have data, you just do not know if the prediction is correct or not. So the idea is combining some experimental data, which are easy to get, and those data could be like MR data. In my lab, we're also interested to using infrared data combined with structural prediction. And this, for medium-sized proteins, that will help greatly because we're so far behind the DNA sequence. We had lots of protein have DNA, but we have very small fraction has structure. And the computation and the minimum data could help to break this barrier.
Thank you again.
Thank you.